I like to tell you about our work on how microbes sense and respond to their environment on a structural level. And you can see this on the screen. So this is E. coli undergoing chemotactic behavior. So the cells can sense and respond to their environment and move toward the sugar source here at the bottom of the slide. So in the greater context, we're also interested in understanding how the cells not only sense and respond to their environment, but also how they structurally adjust their morphology when they encounter different environments. For example, when they enter the dormant stage, like viable but non-culturable stages, where the cells shut down or drive down their metabolic activity and change their morphology, or when they enter a biofilm state, or when they invade a host. And I'm also interested in how bacteria, when they undergo these morphological changes, how do they react to their environment, like environmental assaults like phage attack. But let's start with chemotaxis in the model organism of E. coli. So E. coli are rod-shaped bacteria. They have multiple flagella distributed all over their cell body. Now, if all of these flagella spin in the counterclockwise direction, the cells swim smoothly forward. You can see that on the nice movie from Howard Berg's lab. They swim in these straight runs. Now, if one or more of these flagella start turning clockwise, the cells stop. This bundle dissolves, the cells stop, and they tumble around. Now, by controlling the duration and frequency of these run and tumble phases, the cells can move up attractant gradients or swim away from repellents. Now, this behavior is actually quite simple. There's very few characterized core components. For the chemotaxis system in E. coli, there's only 11 proteins involved. So we have the chemosensory system, which consists of the chemoreceptors that are located at the cell pole. They form huge clusters together with two other proteins, a linking protein, KW, that helps hold these arrays together, and a histidine kinase. Now, this histidine kinase is controlled by the binding state of the receptor. So when attractants and repellents bind, they control the activity of the kinase. The kinase, in turn, phosphorylates a shuttling protein, QY, which then goes to the flagella motor and changes the spinning direction of the flagella. Now, chemotaxis has been studied for over 70 years in detail, and we know a lot of information about the system in E. coli. And we know crystal structures for all the core components that I mentioned. So here you can see the MCPs, the receptors. In E. coli, they form these trimers of dimers in the crystal structure. And then this is the histidine kinase. The histidine kinase is a 5-domain protein that homodimerizes. So we have a structural core that consists of the P3, P4, and P5 domains. So the P3 is the dimerization domain, the P4 is the kinase domain, and the P5 is the receptor binding domain. And there are two other domains attached via flexible linkers. It's the P1 domain that carries the histidine substrate, and the P2 domain, that's the binding site for the shuttling protein. And the third partner is the linking protein KW, and here you see a crystal structure together with the receptor binding domain of the kinase. You can see that it's structurally quite similar. It's a small protein, very similar to the P5 domain of the histidine kinase. Now, we know all this information about these structures, but when I started working on chemotaxis, we had no idea how this puzzle fits together, how these arrays form, and therefore also how they function. So the way we address these questions is by electron tomography. Now, how does it work? First, we freeze our cells. We don't have to prepare them in a traditional way, so we don't need to stain the cells. We don't need to dehydrate them or plastic embed them. The only thing we do is plunge freeze them in liquid ethane. And this freezing process is so fast that not even water has time to crystallize. So everything is stopped in motion, and the structural preservation is preserved. And now the second part is we do tomography. Now when you use transmission electron microscopy, you have to shine your electron beam through your sample, and you collect two-dimensional projection images. Now, when you look on the slide here, and you see your bacterium of choice, in this case it's Mickey Mouse, and on the bottom you can see the projection, or the two-dimensional projection of Mickey Mouse. Now, you wouldn't recognize this three-dimensional object, right? We want to have this three-dimensional information, and so what we do is we don't only take one 2D projection, we take a whole series while we rotate our sample in the microscope. So we collect hundreds of images and then computationally back project them in the computer. Now, here's an example of such a cryo-electron tomogram. This is a Vibrio cell. And we're going to go through this volume, and you can see we can look at all the components of the cell. We can look at the cell envelope. We can look at the chromosome, the ribosomes, cell attachment structures like pili or the flagella. And we can do this with individual cells at macromolecular resolution in 3D. Now, back to the chemoreceptor array. So here's a side view of such a chemoreceptor array. So the dense layer on the top is the inner membrane where the receptors are embedded. And perpendicular, you can see these thin striations, which are the chemoreceptors protruding into the cytoplasm. The lower stripe that you can see here, that's where the linking protein and the kinase binds. Now, if you imagine the side view is a glass of beer, you first drink out the beer, and then you look at the bottom of the glass. And that's a cross-section of these receptors. You can see they're highly organized. So that these are highly organized hexagonal arrays that are formed by these chemoreceptors. Now, I live in the Netherlands, so here you can see a Delft Blue design. They make them in chemoreceptors now. So you can see this is the real architecture of an E. coli chemoreceptor array. So we see, like in purple, you can see the chemoreceptors, and the kinase is labeled in blue and black, and the linking protein is in green. So if you look, you can see that the kinase and the linking protein form alternating rings, and these rings are interconnected by the dimerized histidine kinase. And if you look at this architecture, what I want you to notice is that we have 6 rings of the kinase and the linking protein surrounding one that is empty. So in reality, this might not be empty. It might be filled with fully or partially with only the linking protein, but it's free of Ka. So when I started my own lab, we set out to understand not only the structure of this array, but how does it function? You want to know what happens when attractants and repellents bind. How does this thing work? So what we did is we used genetic mutants that locked these arrays in very distinctive kinase modes. So we have either kinase activating or kinase inactivating strains, and we collect EM maps. Now, these EM maps are great because they're from in vivo arrays, but the resolution is not quite high enough to really see atomic detail. But we can team up with molecular dynamics flexible fitting methods, as done by our collaborator, Dr. Keith Cassidy from the late Klaus Schulte lab. And so he built an all-atom model of 2 million atoms, and he simulated and fitted them in our EM maps to simulate the activation states. Now here, you can see a movie that morphs between the receptor fit into the kinase activating state, and then we morph into the kinase inactivating state. So we can see that when the receptors undergo a change in activation, we see a whole change in the whole trimer. So it's not only in the dimers that the changes occur, but it's the whole trimer that changes conformation. So our current model on how chemotaxis in E. coli works is that we have here the kinase inactivating states. The receptors are zipped up, and also the kinase, the flexible domains from the kinase, are bound underneath the array in an unproductive manner. Now, when the kinase gets activated, the receptors open up, and in turn, then also change the conformation in the kinase and release these two domains on flexible linkers, which allows the capturing of the shuttling protein and the transfer of the phosphoryl group, and it can be sent off to the flagella motor. Now, when I started working on this about 10 years ago, I started not working on E. coli, I worked on Caulobacter crescentus, and the reviewers were very critical. They said, well, you know, who really cares about Caulobacter? We want to know how it's in the important organisms like E. coli or Tamotoga, which is a big important bug for crystallography. So I set out on a long journey and imaged all of these cells or strains that you see here marked in blue asterisks. So I walked down the phylogenetic tree, also in the archaea, and we could show that all of the arrays that we've imaged so far, there's more now than you see on the slide, all of them, the receptors are packed in the same way. They're all hexagonal arrays that have a very distinctive architecture. But are they really the same? So we took a closer look at our new favorite model organism, Vibrio Cholera. And at first glance, here you can see a slice through a tomogram on the left and a subvolume average on the right next to it. So at first glance, they look exactly like the E. coli array. So we have this hexagonal array with the same spacing between the different hexagons. The receptors fit in quite fine. But when we looked at the composition of the base plate, that's where the kinase and the linking protein bind, we see these distinct differences. So while in E. coli, the stoichiometry of the linking protein and the kinase is very conserved. We have this 1-to-1 or 1-to-2 ratio, depending on if the empty hexagons are filled by the linking protein. In contrast, in Vibrio, and this is collaboration with Simon Ringgaard from the Max Planck Institute in Marburg. And there we could show that the stoichiometry is drastically different. So in Vibrio, we have much less kinase in the array. And on top of that, there are additional proteins in Vibrio that integrate into the base plate. So there's a protein called Kiwi and also PARP. Now, what does this mean? So you see these labeled receptor trimers in red. So if you look at E. coli, each trimer actually connects to a kinase. Whereas in Vibrio, all of these red ones are not in direct contact with a kinase. So we need this ordered packing and this tight regulated packing to allow these receptors to transmit the signal through the array to activate a kinase. Now, does it also have structural consequences? And yes, it does. So in E. coli, these arrays, the kinases staple these arrays together. So they're super stable. So you can lyse the cells completely, and you will still have these beautiful conserved arrays, which you can see here. In contrast, on the right, you can see such an array in a slightly lysed Vibrio cell. And as soon as you just gently lyse these Vibrio cells, you can see these arrays fall apart. So they're also structurally different than the E. coli arrays. Now, does this have any meaning if you look at it from the environmental standpoint? And for that, we looked at this viable but non-culturable cell state in Vibrio. Now here you can see my postdoc, Susanne Brenzinger, when we went out to collect natural water samples. And you can see we're in the Netherlands. You can see a little windmill in the back. That's the proof. Anyway, so we cultured typically grown overnight cultures in LB, which you can see here, the nice Vibrioid-shaped cell. And then next to it, you can see such a viable but non-culturable cell state in Vibrio. So these cells change their morphology dramatically. So the inner membrane rounds up, the outer membrane sloughs off, so they look like they have like a fried egg morphology. The chromosome condenses. You can see this outlined here in pink. But if you look at the chemoreceptor array marked in yellow, we don't see any structural differences. So just zooming in a little bit. So here you can see it again labeled in yellow. And there we can't distinguish the structure of these arrays. But when we look at the proteomic changes between overnight cultures and the VBNC state, we see a dramatic change in the composition of the receptor. So these are all chemotaxis proteins that go to this array. So Vibrio is fantastic and has 3 different systems, but these are just the proteins from this array. And most of them are the receptors. So you can see they change quite dramatically. So we think that in the environment, it helps Vibrio that these arrays are not ultra-stable because it allows them to switch out the receptors when they change the environment. And then they always have the right type of receptor to encounter or thrive in different environments. Now, the question we're currently working on is, do these dramatic morphological changes have any effect also on what happens to the cell when they're undergoing phage attack, for example? And so this is a very, very recent work from the last few weeks. So we started working with a Vibrio phage called ICP1. This phage is very typically isolated from cholera patients in Bangladesh. There are some beautiful studies on this virus, on this phage out there, but structurally it was not very well characterized. So we set out to first understand the structure of this virus, and you can see that spinning here. So it's a typically icosahedral phage head, this nice tail, and a very characteristic base plate. Now, if you look at these EM pictures next to the structure, you can see the different stages of the phage from just a phage prep. So we can see the full unattached virus where the phage head is still packed with DNA. You can see next to it phage heads that are half empty, so were frozen right in the middle of DNA ejection. And also we can see on the left an empty phage head. So now when we add these phages to Vibrio, to these VBNC cells, we can see them attaching here. And even the fully attached phages, none of them eject their genome. So they're all filled. And most of them seem to be just too far away from the inner membrane to even reach it with the needle. But in a small inset, you can see, in some instances, we do see that the needle reaches the inner membrane, but still we don't see DNA ejection. And if we do a phage assay, we can see in the presence of these VBNC cells, the phage load in the environment is dramatically reduced. So even if these VBNC cells will never recover and start replicating again, they might still help their sister cells that are still around by reducing the phage load. Now, the last big project I want to just briefly tell you about is Vibrio infection of zebrafish larvae. So this is a recent project we just started. It's a very brave PhD student, Jamie Depalto, who took on this huge project. And we want to understand, how does Vibrio change when they infect a host? Now, this is a very, very challenging question for cryo-electron tomography, because the samples that we need to insert in the microscope have to be 500 nanometer or thinner. And a zebrafish is certainly bigger than that. So at the moment, we address this question by a different type of electron microscopy. It's called block phase SEM. So it's a scanning electron microscope. It has a diamond knife built in. So what you do is you have an embedded zebrafish larvae, and then you take the diamond knife, you slice off the top. You take an image, and you keep slicing and imaging. So here is just to show, this is such a zebrafish larvae, and on the bottom you can see a movie of an infection of the gut. So it's a Vibrio strain with a GFP marker. So we have a really nice infection model here. And now, so here's the first block phase SEM results. We actually have a lot of these movies. We have run the microscope for 7 days straight. We have up to 650 micrometer blocks that we have now to analyze, so I don't have the full analysis, but I want to show you how this looks like. It's just a small segment. And in the middle of the lumen, you can see these little dark dots. These are all Vibrio cells. And on the upper right corner, you can see a dying cell from the host. So we have a lot of data. We still have to figure out how to analyze these massive data sets. Now, the last minute, I just want to tell you about my hope for showing our data to the public. Now, it's great to tell you all about our research, but we also want to educate the public. We want to have school children learn about the inner life of bacteria and show them what we can do. And our technique is so visual that I think it's a very good method to convey this to the public. So we have currently a pilot project running together with the first microbe museum in Amsterdam called Micropia. So it's if you have not been there, it's a fascinating museum to go to. It's a program that was started with where psychologists, microbiologists, sound engineers, and artists got together and created an amazing space where kids can go to. There's lots of microscopes to play with. It's an amazing experience. So I hope if you come anywhere end of the summer, you will see movies from our work on exhibition. And the driving force of this is a master's student, a media technology master's student, Vera, you can see in this picture. So with that, I want to thank the people who actually did the work. So we have Vera Williams, who's spearheading the Micropia project. Then we have Susanne Brenzinger, who worked on the persister cells and the phage project. She escaped her own picture frame, is now bugging a PhD student, Eveline. Then the whole chemotaxis work, the high-resolution structures were done by my fabulous PhD student, Wen Yang. Jamie Depolto is the one who's spearheading the Zebrafish project. And very important for us, Christoph Diepholder, he's an operator in our microscope center. So we have the Niesen, the Netherlands Center for Electron Nanoscopy. So it's an open center. So if you now all want to do electron tomography, you can actually access such microscopes in the center. And they have expert staff to help you get this data. Thanks to my fabulous collaborators, Sandy Parkinson, who's done the genetic mutants of E. coli for the Chemotaxis project, Keith Cassidy, who's done the molecular dynamics for us, Anna-Marie Meyer and Björn Koch help us with all the zebrafish work, because I've never worked with anything more than one cell before. Simon Ringard and Timo Glatter from Marburg helped us with the Vibrio proteomics project, and Bram Koster and his group help us with the block phase SCM. And I want to highlight the scientific artwork that I showed you here. Scientific illustrations, I think, are so powerful to convey a message. So I want to highlight Frank Santarello, who has a poster here, and this overview drawing is from him. So make sure you go to his poster. I'm sure it will be beautiful. And I want to thank the funding sources. And thank you for your attention.